Start getting people filter in. Yes, we've got a few. Welcome for those of you who've joined bang on time. Thank you very much. We're just going to wait a couple of minutes just for people to filter in. Maybe just as a little bit of an icebreaker for those of you who were able to attend the episode 1 of our DOE Office Hours. If you could just drop a little message or emoji in the chat, just so we can get an idea of who's here, that'd be super helpful. Thank you. And we'll kick off in just a couple of minutes. We've got a few returning visitors. We need some lift music or something maybe whilst we wait.
Exactly. Oh, it's good to see we're not alone, Sid. Have some viewers. Very welcome.
Cool. Okay. I think we've got a fair number of people here. So let's get started then. Cool. So welcome everybody to the second installment of the Synthace DOE Office Hours. We're going to be joined today by Dr. Michael Sadowski, Nathan Hardingham, and I'll do full introductions for them all both in a second. They're going to be talking through sort of the application of DOE. So for those of you who are able to attend the first session, we covered the basics of DOE. This session today is going to be a little bit more focused around actually applying it to your own work. Before we dive in to introductions and then the real meat of the webinar, just a couple of housekeeping points for everybody. Please do just let us know in the chat or in a private message. You should be able to send both of those if you do have any technical difficulties or anything as the webinar is going along. So sound issues, that type of thing, please just shout. The webinar will be recorded today and you'll get an email right after the session with the recording. So please feel free to share that with colleagues or anyone else in your teams who's maybe interested to learn a bit more about. I do want to highlight as well that as with the first session, for those of you who are there, this is a really interactive session. We want this to be as open a discussion as we can. We've obviously got some pre-selected and pre-submitted questions that Nathan and Sid will be trying to answer today. But yeah, keep it coming in the chat, ask questions. I'll be sitting in the chat, moderating that and trying to answer any questions as well. Will be some time at the end to answer some sort of live questions that will be submitted in the chat. And there will also be a follow-up survey as you finish as well. So once the webinar ends, you'll get a little survey just on your feedback or thoughts on the webinar. And we do love to hear from how these things are received, and our marketing team will definitely thank you for your feedback. So if you do have some time, it would be really appreciated if you could just fill that in. But so I guess without further ado, I'll just quick introduction to our speakers today. We've got Nathan, who sits in the customer success team here at Synthace. So he's a customer success scientist and is basically working day to help support and enable our users of our platform to do DOE. So he's real at the coalface working to enable scientists to implement DOE in their work. So very lucky to have him join us today. And then we've got Dr. Michael Sadowski. You'll hear us refer to him as Sid. He is Sid to everybody, I think, at this point. You might remember him from the first session. He's the Director of Scientific Software here at Synthace, real DOE evangelist. Has got many years in DOE and DOE consulting, as well as all things machine learning, AI, and biology optimization. So that's enough talking from me. I will hand over to folks to kick us off.
Cool. Yeah. Thank you I'm happy to be going back to back with you today, Sid. And for those of you who don't know Sid, I know maybe some of you saw the previous webinar, but honestly, he is one of the brilliant minds of the DOE world. He won't admit it. He's my go-to for DOE questions. I've pestered him so much that I'm surprised I am not on his block list yet. Seriously though, delighted to be sharing the podium with you today, mate. So let's kick it off. Sid, first question, how can I use DOE to optimize automated lab processes? For example, colony picking.
Thanks a lot, guys. One step closer to being blocked with that intro. I told him not to do that. Okay, so yeah, I need to quickly preface what I say by saying I've not really done colony picking, so I'm no expert here. I have talked to a few people who have done a lot of it though, so hopefully I'm not completely away from reality here, but the idea is just to give you a flavor of how some of the things we said last time play out in actually a relatively simple kind of example. So hopefully you'll get something from that. So yeah, if you were in the last webinar, you might remember this slide. This was my sort of commandments of starting your DOE kind of slides, the first golden rule is always that you need to work out what on earth it is you're trying to achieve. What are you trying to do? And then you can start thinking about, okay, so how much is it worth to get there? How much resources, how much time, how much effort is this worth? And how am I going to actually know when I have got there? Is my measurement up to scratch? What's it going to tell me? So first off, let's quickly revise colony picking for any of you who don't know anything about it. So here's an example of a couple of plates that you might encounter. So basically the idea is you've got these plates, you've grown some bacteria on them. They should ideally grow into individual discrete colonies, which then should be clonal. So they should all have the same genotype essentially. And then you're going to go and pick one of these to then take that specific genetic background, those constructs to go and do something else with. And so people do this by hand, of course. But we also have these interesting machines that automate it. So I think this is like a retrofit on a conventional liquid handler, like a TCAN or something like that. There are dedicated machines to do this, but this was the best fair use picture I could find of one. And so basically what you've got here is you've mostly got a couple of key components. You've got this arm in the middle that actually has a holder pipette tip, goes and picks up a colony and then puts it in a plate, as you can see down on the right, the 96 or the 6-well plate there. And then there's also a camera, which is the first thing that happens, which images the plate and then tries to figure out where the colonies are. So if we think about optimizing in DOE terms, this gives us a few places to think about factors. So one obvious area, which I'm not going to cover here because it wasn't really in the original question, it's a bit more complicated, is the question of actually how you pick up the colonies in the first place. That's extremely important. And actually, I would definitely want to try and think about that when I'm doing my optimization. I'd want to consider the detection algorithm and how the physical part goes, ideally. But we're going to stick just to the physical part for the moment. So that's more like, given, let's assume that the detection has worked and it's found a colony, are we actually going to successfully transfer enough of that colony into our agar to then grow? And so there's really three main parameters that the questioner asked about. There's the sort of the speed the tip goes down. There's a sort of a detection system that tells you when you've hit the colony, and so you don't want to go too deep, otherwise you'll just pick up loads of agar and not enough bacteria. So that's a pressure. And then there's this mixing vigorousness, which is how well you transfer it into the 96-well plate. So those are the three main factors we're going to think about. And we need to think about also, like, how are we going to measure success? Now, there's a lot of different ways you could do that, but the best suggestion I had was that you'd probably go off and then extract the DNA, amplify it, and then measure how much you'd got and how pure it was and that kind of thing. That involves probably doing a lot of mini preps, which can be a little bit arduous. So that sort of feeds into our question of how many runs we can do. Sort of roughly, it's anything up to 96 a day is possible. You're in the tens region. So that gives you an idea of the effort you'd need to actually do your response. One other thing that isn't a factor, but I wanted to bring up here, which is really key in this kind of situation, is we've talked about wanting to optimize, say, for how much DNA you get out, which is our response. But the other question is what actually is the scope of this? Like, different organisms, different aspects of genetics, different growth protocols could lead to very different colony formation. And so that's something else you'd really want to consider. You can see there's quite a lot of differences in the sorts of plates you get out, which will affect mostly the detection part, but I think it could also have implications for how you actually go and transfer the DNA. So that's something you need to think about. But for our purposes, let's assume we're using a single well-defined protocol against a very narrow range of possible organisms. So they're going to be quite consistent. So if we go back to our commandment slide, we've now pretty much figured out a rough idea of what we need to achieve, what it might cost, and how we could measure progress. So we want to optimize for successfully hitting a colony. We're just going to do it for a consistent process. I'm going to arbitrarily say we've got a bit of time. Let's say this takes a few days. We'll maybe want to spend a couple of weeks doing it. So that probably buys us about 3 iterations. It doesn't have to be exact, but just to give us an idea of the scope here. Probably the biggest limitation is the assay. We might be able to get up to 96. Really depends if you've got any extra automation there. It can be a bit of a slog to do that many. So we're talking, you can do up to, say, about between 48 and 96 runs. So it's not a big limitation. So I then think about another slide that you've seen before, is like, when we talk about iterations, what kind of things do we need? So there's all these different stages you can do in DOE. If you're dealing with lots of factors and you really don't know much about your process, you'd probably start all the way over on the left in scoping to try and figure out what's a decent range for my things, my factors. We might then screen to try and get rid of factors that aren't that important and make the whole optimization more tractable. And then we might end up optimizing. But in this case, we've got 3 factors. We've said the process is very diverse. We can probably skip straight to this bit, which is just to optimize. And then we want to leave a little room for robustness, which is just double checking that what we've got actually is what we think we've got. And then we'd move into this sort of standard kind of thing. I'm not going to this whole slide, but this is then what we would do. So in an optimization context, we would then say, okay, we've got 3 factors. We've probably got a smallish number of levels. We're not too bad on runs, probably up to a 96-ish. So then we could choose the kind of design that lets us optimize. So that would typically be an RSM design or an iOptimal design. We could run it and then do the optimization. And then that might just work. We might have to try again because we've made a mistake and some of our settings are way off. And we've got a bit of room to do that if we're allowing potentially for 3 iterations. So that's really it. That's a sort of a whistle-stop tour of how it might play out in this situation. I hope that's helpful.
Yeah, it's an interesting example, actually. And for me, at least, I feel like colony picking feels like a little bit of a niche example, which I quite like. And it's different to what, I'll speak about in the next few questions a lot, which is optimizing cell growth media. And in that case, you're often just optimizing components of a mixture, which is often quite easy to do. All your factors are easy to change. But with something like this, maybe it's difficult to identify, what your factors are in the first instance. Is a bit of a challenge. So yeah, I suppose my sort of question to you, Sid, is that a challenge? What's your sort of biggest challenge with this sort of maybe a niche process when you're starting off?
Yeah, in this specific example, I think it's relatively straightforward, but you can easily encounter situations where your machine does a batch of things at once, right? And one of the settings only applies to a batch. It doesn't apply to a single thing. Like you can't change it per individual instance. That's a very common thing in this case. And that's what you'd call typically a hard to change factor. It didn't really come up in this example, but it's very common. And that requires you to do something like a split plot design. I think there's some stuff later on where we can build on that a bit more, but that's probably the most obvious one.
Cool. Should we move it on, Sid?
Yeah, let's go. All right. Whoops. If I can hit the right slide. So question 2 was about cell growth medium optimization, basically. So I think, the particular examples were things like stem cells, disease cells, for obvious reasons. Yeah. Can you help us out with that?
Sure. I can definitely give it a go. Yeah, we're moving on to talk about cell growth media optimization here. In my mind, this is a perfect application of DOE. No matter what cells you have, they're going to be incredibly sensitive to the concentrations of the components of your media. And I know from experience, we're often using a one-size-fits-all media from our manufacturer, and that can give us mediocre growth of our cells. But it's not just the media. Honestly, it feels everything impacts your cells, from temperature, shaking speed, maybe even your star sign. But with media optimization, there's really unlimited factors to consider. And even small increases in yield can be really beneficial for some companies. It doesn't have to be yield, it's just an example. So when we are starting off a media optimization campaign, here are some tips and tricks, some things that I learned along the way. So first, think about what you know. I'd probably do this with any DOE campaign. It's really important to make maybe a fishbone diagram, or we also call it Ishikawa plot, to consider all of the factors you could test in your DOE experiment. The next thing, of course, is forget everything. Cell growth media, it's like Times Square on a Friday night. There's so much going on, and we may think that we know the best concentrations of every component that will give us the best cell growth. But in actual fact, there's going to be huge interactions between your media components. Some components are going to have far more significant effects than you may have anticipated, and some are not going to make a blind bit of difference. So what are you going to do about it? So first, one kind of bit of advice is do a scoping experiment or scoping experiments. Invest your time in doing a trial experiment before you run a big DOE campaign. The reason being, whenever you run any experiment, particularly with cells, we're talking about cells here, is you're going to see a lot of noise. So in your experiment, noise is the unwanted variation or error in your data, and it makes it harder to see meaningful effects. In a cell-based experiment, you'll see a lot of that noise. Cells have variations in metabolism, responses to environment, that sort of thing, and they're highly responsive to those environmental factors. So to give an example, I suppose to do this, you could set up a condition with high concentrations of all your media components, low concentrations of all your media components, and maybe a middle ground. Perhaps this is your day-to-day media, and test those out with a few replicates each. Now, this is cool because you'll be able to see whether actually some media components, whether they're lethal to your cells at high concentrations. Now, we've actually seen this firsthand. Even if we think that the upper limit for our media, even if we think we know that, actually, when you put them together, the interaction between certain media components at high concentrations can be lethal to your cells. But what's interesting as well is if you do replicates of these separate conditions, you'll also get a feel for the noise or variation in your system. And hopefully by doing this as well, you might also find a starting point for your factor levels, which is something that people often find tricky. They don't know where to start. So a question we get a lot is, should I prioritize runs or replicates? It feels like a simple question. It stumbles a lot of people. I like this question when I get it. For example, what's better? Let's say 150 runs or doing 50 runs 3 replicates. In my experience, replicates are far more important for cell-based experiments. Now, if you're running an assay where you wouldn't expect much noise, sure, go with more runs. But if it's a cell-based experiment, you need replicates. And really the reason you're going to do this is because the replicates will average out those random fluctuations or noise. If you have a lot of replicates, you're going to be impacted less by that variation. It's as simple as that. Then, once you're happy with that, maybe think about how you arrange your runs. And we're talking about randomizing, blocking, and controlling. In terms of randomization, often people are a little bit scared to randomize the runs in your plate. We like to know where things are as scientists. And to be honest, organizing things perfectly can make it easier to record your data. It's difficult to execute when things are randomized. If possible, find a software solution that allows you to randomize properly. I'm not giving any shout outs here. But different parts of your plate are going to be subject to different environmental conditions. Particularly the edge wells, we see that they're more exposed. And by randomizing, you're going to make sure that any variation that you will see is going to be evenly distributed across the conditions you're testing. Yeah, don't be tempted to organize your runs like a filing cabinet. Randomization is really power here. Consider doing blocking. The second point, we're going to talk about that a bit more later. So just remember that one. But blocking can help minimize the impact of your known sources of variation. And then the final one here is controls. In particular, when you run, Sid was talking about iterations of experiments. When you run iterations, keep some conditions in common between your last iteration. We might see that people have drastically different results between their cell-based experiments 1 week and the next. And this is a way of sort of sanity checking whether there's been some catastrophic environmental event that might have affected your cells on that particular day. And finally, do it again. Do it all over. If you think DOE is going to be one and done, have some bad news for you. But DOE, and particular in media optimization DOE, you're going to have multiple follow-up experiments to truly understand your system. When we call these iterations, you'll hear us use this term. But each iteration is going to build on your understanding of the last. I suppose to give an example, maybe if you found some factors that aren't, you might want to fix their levels in follow-up experiments. But if you found some factors that have strong statistically significant impacts, then tweak those levels, investigate that further. Maybe you found increasing glucose concentration gives you a better yield. So keep climbing up that hill. If that's what you want, keep climbing up that hill. And maybe continue perhaps with a de-optimal or fractional factorial screening design. But let's say you're climbing up that hill and you found your peak, you think you found your peak, and you believe you found where the impact of your factor begins to plateau. Perhaps it's time to build an optimization model, maybe an I-optimal or a central composite design. But really the specifics of the design is not important here. The point is keep going, build on your last experiment. And the last point to wrap it up, don't be afraid to break your system. And what I mean is push that factor level to the extremities. Find the point at which your system simply cannot tolerate any more of your factor. And this is really valuable information because there's no point thinking that your optimal temperature is 35 degrees, when it happens at 37 degrees, your cells will die. Your conditions are really only truly optimized if they are robust too.
Fantastic. Thanks, Nathan. Yeah, I did have a question to follow up, but I think in the interest of time, let's plow on for the moment.
Sure. Let's do it.
So do you want to move on to the next one?
Let's do it. Cool. Sid, yes. What design would you use to screen for relevant factors in a cultivation experiment? For example, maximizing recombinant protein yield.
Yeah, nice. So this is a great trip down memory lane. This is actually something this is where we started as a company, way before we became a software company. We did a lot. This was our big thing, was DOEing, expressing difficult proteins and that kind of thing. So yeah, and here's a picture from that time. It's just a little schematic just to help people understand what's going on. So yeah, we've got a bug here, which we're going to put some genes in to help it make whatever it is we're interested in making. And it's going to take some kind of feedstock in, and that will produce a product. So you have to grow them up, feed them with whatever it is, and then they should, in an ideal world, produce tons and tons of your product and make you very rich. So the key thing, our big deal here, our big insight was that you should think about the whole thing, ideally. So it's very common for people to try and optimize these things separately, but the problem in that case is you're assuming that they don't interact with each other. And actually, as our experience repeatedly showed, genetics and environment interact a lot. Who knew? In fact, what we did was we did a big DOE across the whole process and carried factors from one stage to another. For example, we would consider things like the growth medium, to build on what you just said, and how long you're doing your incubations, your growths, that kind of thing, how many cells you're putting in, whether there's any passaging. There's tons of factors to do with the genetics, the specific protein you're encoding, how you're encoding it, what plasmid it's on, how it's being regulated, so it's promoter and terminator and stuff. Whether there's any ancillary proteins need to go in, loads of things potentially there. And then there's things to do with the background strain itself. What strain did you take? Did you do anything to it? These are all things you might consider. And obviously you can't optimize every one of these. You have to go through a process of choosing. But the main thing is you really want to think about where are the big interactions going to be. And it's usually there's bits in the medium that interact with some of the genetic things. Temperature often comes out. And so, yeah, we built a process along the top of that. Here's a sort of a depiction of it. The details don't matter too much, but it's more or less a recapitulation of what I just said. And that's the interesting point here is, while this was innovative for biotechnology, it's not at all innovative for DOE. In fact, it is classical response surface methodology DOE. So we started with simple fractional factorials to screen out factors. We then, once we'd figured out what factors really mattered and what interactions, we then made the design more dense and fixed some factors. Then when we were at a point where we thought we could actually optimize, we'd do a central composite design and then get as much out of the system as possible and finally do a little bit of robustness. And that worked extremely well. So this is a case study we've done flogged to death in a way, but it's a brilliant one. It was I think it's like a 4 or 5 week campaign in which we got a huge increase in expression of a human enzyme in E. coli, which is not the easiest thing to do. And so, yeah, you can start over there on the left with the blue peaks. You see there's one that's okay, and the rest are just more or less nothing. It's 1 or 2. Then as we go up the iterations, the averages start to go up, and so do the peaks, until we get to the purple ones where we found some really good ones. And then finally, our robustness at the end showed that there was some variation, but generally speaking, we got pretty good, consistent expression. And this was all done, as I said, with the classical sort of DOE type stuff. So that's the nice thing. This is one of the reasons we believe in DOE so much, is we found that even though this is a big, complicated process, DOE, and just, you don't have to take the fanciest, best DOEs. You can just do a normal one and it works well.
Yeah, I love this graph, Sid, because it's something I actually use to explain to people the power of iteration. It's something you talked about a lot, and it's a really nice demonstration of that. I did want to pick your brain actually about this sort of accounting for factors across different stages that are quite independent. Do you want to cover that now, or maybe if we have time in the Q&A session, or should we chat about it now?
I think I've accidentally said the main thing, which is, I could just quickly reiterate it. It's basically about, it can be a hard thing to administrate. It takes a lot of orchestration, but if you don't think about the whole process, you run a big risk of things interacting that actually you're completely blind to, because you're treating them as that you're assuming they're independent. So yeah, it's a big burden of orchestration. Can't really go into what it is now, but it's lots and lots of planning and difficult execution, but it's worth it. Okay, so yeah, so perhaps the other end of the scale, all relatedly actually, we had someone who comes from quite a small lab without many resources, mostly doing stuff manually. I was interested to say, is DOE just too hard for me to do? What do you think?
Yeah, I love this question. DOE experts, I don't profess to be one. DOE experts often don't want to admit there's a huge barrier to doing DOE, and this being one of them. We often speak about DOE, or myself included, in an automation context. Running experiments automatically allows you to test a lot of variables and factors. So what about limited resources? And I suppose that's a manual execution, if you like. What about manual DOE? Can you do it? Is it possible? And I would say absolutely. I'll give an example. Maybe you've got a simple biochemical assay. I've got an example of one here. And let's say we want to optimize the levels of each component for whatever output you're measuring. We'll probably have an enzyme, a substrate, and a buffer in our assay. So let's say we've got this substrate, I've said 5, 10, and 15 micromolar. An enzyme, 5, 10, 50 micromolar as well, and then a buffer at pH 6, 7, and 8. So imagine we chose the simplest design possible, a full factorial design. And that's where we test every single level, every single combination of every single factor at every single level. And to work out actually how many runs that is simply the number of levels to the power number of factors, in this case 3 to the power 3. And if you're good at mental maths, you'll know that's 27. So that's 27 runs. And with a rack of pipette tips and a free trial of jump, I'd say that's pretty achievable. So the question actually goes a bit more specific than this. I'm demonstrating here. I think you still can do a simple assay in a DOE context with actually not going into too many runs and actually therefore too much cost. But actually goes into more specific context about needing to split out experiment into multiple days. So in actual fact, often people with automation will want to do this anyway, and we call this blocking. Until a couple of years ago, blocking to me was something I do on the football pitch, but turns out we do it in DOE too. And blocking really is something where you set up your experiment to account for a nuisance variable, something you either can't control or even just don't want to control. But it's something that, irrespective of that, is likely to affect your results. So remember those 27 runs from that experiment. Let's say Sid is kindly going to help me out, and he's going to pipette some of the runs for me, and I'm going to do the rest. Problem is there's a source of variation there. Sid is far better with a pipette than I am, so he's likely to be more accurate with a pipette than I am. Let's assume. In this case, the person pipetting is the nuisance variable, and I'm sure you are better than me. We're not interested in who pipettes most accurately. We're interested really in the performance of our assay. So what we're going to do is we're going to block by the person pipetting. We're going to arrange our experiment, our runs, in such a way that we won't confuse your mad pipetting skills, Sid, with how our factors affect our response. So in your DOE software, for example, this is how you're going to do this. You could set a blocking factor. And your values, I put this at the bottom, your values might be Sid and Nathan. In this case, we'd set the number of runs per block at 14, simply because we've got 27 runs. So we're trying to split that between two as evenly as possible. So the design, and not you, the design is going to do this, the software is going to do this. It's going to split out the runs into blocks of 14 or as close to as possible. In this case, it's going to be 14 for me and 13 for Sid because we've got 27 runs. And then the software is going to strategically organize the runs between us such that we've got as even as possible combinations of those factor levels. And so the reason it's doing this really is to mitigate risk that you'll confuse the effect of different people pipetting with the effect of a factor on our response. And that really it's just a case of letting the software arrange your design into blocks. It's not a flawless method by any means, but it reduces risk of what we call, and it's a good mitigation. But so I suppose something I get confused between, Sid, to be completely honest, is hard to change factors and blocking, and what's the difference? Are they the same? And I was actually wondering if you could clear that up for me, if you don't mind.
Yeah, sure. Just as a comment, there's a reason I left anything experimental at the earliest possible opportunity. I'm terrible pipetting technique. Yes, the difference is, I think you pretty much summarized blocking really well there. Blocking's when you don't care about some factor that might interfere with your experiment. A hard to change factor is a factor that might affect your experiment that you do care about. And so the way you deal with them is somewhat similar. You have to make sure that there's no confounding between the blocks and the factors, or the hard to change factor and the other factors and level and effects you're interested in. But you also do a bit of extra stuff. So it's a so-called split plot design. It usually adds it adds some extra blocks, if you like, that would be more than you'd normally have in a purely blocked experiment, to help you distinguish between the effect of your hard to change factor and anything that happens because you've blocked, basically. That's it in a nutshell, really.
Yeah, that makes a lot of sense. I mean, I think in essence, like, blocking is actually quite a simple concept when you think about it. Cool. So Sid, how can you determine whether a factor is important? I like this question already.
Yeah, me too. Although it's so broad. It could take up a lot of time, but I'll try and be as brief as I can. Essentially, the one thing I want to go in with is to ask, what's the motivation for this question, right? And typically it's because you've probably got quite a lot of factors and you're screening and you want to try and stop varying some. So I've converted it almost into the question of, when is it safe to do that? What are the considerations? So first off, there's different kinds of factors. There's different things you're varying in your experiment that you then may or may not fix ever, basically. And the key distinction is, so you've got, on the left, I've got experimental factors. That's your normal factors. They're the things that the dials you're trying to find settings for, which you're basically going to fix those settings probably in the real case, and they'll just stay there, no problem. But you also have this idea that there will be things in the real case that vary that maybe you can't control. That's this idea. So when you're at the experimenting, you often can try and control those things and see what effect they have. So temperature is a very key example. How pure your source material is, what effect different equipment has. Those are all different things that you might do here. And similarly, referring back to my sort of colony picking example, this idea of this is more like, okay, so there's something in here that my process has to cope with. I'll always know what it is ahead of time. I've got to do some colonies from this particular process of this organism, but you still have to, it's still different. It still changes between instances of the process. So, how much DMSO there is in your assay as an example, that often comes up. What strain you're working with might be another one. So these latter two are factors where probably want to keep them in the whole time. It's really only the first experimental kind you're thinking about fixing. Another question, even within those, is why are you investigating it? There's different reasons you bring things in. One nice thing about DOE is you can do more, so you can investigate things you're not so sure about. If it's one of these kind of long shots, maybe you do want to think about it. But on the flip side, usually there's tuning to be done of any given system. When you give organisms different kinds of nutrients, quite often they have to be balanced with each other. Those kind of things you probably not fix until the last possible moment. And finally, if you're just trying to understand the effect of something, obviously you're always going to keep varying it. The final sort of caveat I wanted to give is, I would always advocate for fixing factors as late as possible. So if this little pretty diagram is supposed to represent the idea that you could think about your experimental process as searching a bumpy landscape for, in this case, the lowest point. You'd probably more naturally think about it as the highest, but doesn't really matter. That's just what the diagram has. If you look at this and you think about, okay, let's say I've done my experiment over here and I found that X2 is quite flat. Okay, there's no slope there, which would mean it's not significant, which would mean I might then think, oh, it doesn't matter. But of course, yes, it doesn't have much effect there. But if I move over here, suddenly there's a really big slope in X2. So out of context, there's no real meaning to the idea that it has no effect, right? The real meaning of that would be flat everywhere, which can happen, of course. But it's a conclusion you don't want to necessarily jump to too quickly. So it's best to start thinking about screening when you're a bit closer to your optimum. So I tend to think, be a bit slow to remove factors. But so let's finally, what criteria would you use apart from the ones that I've raised? Like, a lot of it is this sort of question of significance. That's one thing. If That just means, are we sure that there's a slope there in that variable for that effect? So can we distinguish it from noise? But there's a lot of things that can confound that and make it harder to tell. And one thing that can go wrong is you can have loads and loads of runs and very little noise, and then everything looks massively significant. But you're picking up really small effects. So another really good question to ask is just like, how big does this coefficient look? And it's important to be careful here, because the coefficients, just as they come out of the box, aren't scaled to each other. They're scaled to how you've encoded the factors. So if one of the ranges of the factors is like 100 times another, that coefficient will always look 100 times bigger comparatively. So that's something you've got to be careful about. And finally, yeah, just be careful. Your design will constrain what effects you can reliably detect. So you need to be careful and make sure that that's telling you that you're not dismissing something out of hand because the design can't tell you, rather than the numbers actually are reflecting reality. So I hope that's a reasonable summary that makes some kind of sense.
I really like the image of the experimental landscape.
Yeah, I wish I
want to hang it and frame it. But you spoke about fixing, you used that word fix. I think maybe it's not even immediately obvious when you start doing DOE that actually it's not always that you find maybe a factor isn't significantly impacting your response. And people talk about dropping that as a factor, but it's not dropping it, it's actually just fixing it at a point, right?
Yeah, that's exactly it. It's just, what are you varying, not what are you setting. All of your factors will always have some value. Some of them you won't know, some of them you won't set, some of them you won't change it deliberately. But yeah, that's what fixing really means, is we used to be varying it and now we're not. We're just going to choose a value for it.
Nice. Should we move on?
Yeah. So the next one, more also about cell growth medium, actually, again. So how do we optimize one of those efficiently to get, say, high titer or low cost?
Nice. Yeah, I am really liking these questions around reducing cost and taking that spin on it, reducing runs. I think it's real. And it's the same reason I like shopping on Vinted or eBay. I'm not cheap. I just love a bargain. And just like buying secondhand clothes or something like that, in science, you'll often want a good result at a cut rate price. So I paraphrased, how do you do DOE super efficiently whilst reducing cost? So for most people, reducing cost will effectively mean reducing the number of experimental runs. In media optimization, actually, in that context, when you're optimizing cell growth media, the first thing you might actually want to do is reduce the volume. Reducing the amount of volume of media in each run is actually probably the fastest way to reduce the amount of media components you use, and that will reduce cost. Ultimately, if you're optimizing for yield or something similar, it's going to be relative to the volume of media. If you're in an early scale up, this is probably fine to do. But from there, we've got some other approaches for you. So I'm going to take a really generic media optimization example where you're optimizing for cell yield. There's a few factors that you might want to test here. And there are different approaches to this problem. I'm purposely going to make some sweeping statements and assumptions here because I want to be as specific as possible. So if you can't test them all, let's prioritize. And for those that we don't want to vary, we can simply fix the concentrations of them using that fix word we just spoke about. So here's where you are going to use your experience. I've made some huge assumptions, so just take these at face value. So these are our components we could test. So we're going to cut down on the number of factors simply to reduce cost and reduce the number of runs we need. So from my experience, at least, I put salt type there and I've crossed through it. The type of salt you use is usually not pivotal to your cell's yield, so we can ditch that as a factor. You could even ditch investigating the concentration of salt used. There's a debate in some media whether it's needed at all, but changing the buffer type too often makes minimal difference. We've dropped that one here. And the reason being, I've seen the type of buffer doesn't make a huge difference. You need it there, but the type of buffer doesn't make a huge difference. And it's also rare that you get strong interactions from your buffer and your media components. So you could, and Sid might kill me for saying this, but investigate buffer type as a separate experiment later on. I know that's urging on one factor at a time. You could feasibly do that. Shocked, I know, Sid. Yeah, and also the concentration of nitrogen, carbon sources. So nitrogen like your amino acids, carbon like your glucose, growth factors like EGF. But these are going to be the factors that will probably come out as having huge statistical significance. But having said that, you might just choose one nitrogen, one carbon, and one growth factor source. So we could choose to not factorize the type of nitrogen, carbon, and growth factor source, and rather just pick one of them and focus on manipulating its concentration in the media. And then finally, you'll see in the top right, I've crossed out temperature. It's likely to make a really significant difference, but it's often difficult to factorize because you'll need to separate runs into different plates so they can go in different incubators. So if we're being brutal, we're getting rid of that too. But really what you can actually see that we've done here is prioritize those continuous factors and deprioritize the categorical or those hard to change factors. And really the reason for this is that the categorical factors, like testing 3 different carbon sources, will require you to test all those 3 carbon sources independently. And what's interesting is you can't predict the behavior of a category without testing it. If you have glucose, galactose, lactose, you can't predict the impact of one of those categories without actually testing it. And conversely, if you just had glucose, you could test low concentration of glucose, high concentration of glucose, and have a pretty good idea of what that midpoint glucose concentration would look like. But if, for example, you wanted to measure all of those 3 carbon sources in a full factorial design, you need to test them all. So you might triple the number of experiments you might need to do. We can reduce the number of runs and by deprioritizing those categorical factors if we're being really brutal. So after being brutal with those factors, we've got 6 left. And for each of them, I've added 2 levels. Really, they're arbitrary. But we've got 1 nitrogen source, 1 carbon source, 1 growth factor, 1 salt, 1 organic acid, and pH, which we probably model as having a buffer at pH 6 and 1 at pH 8. So with 6 factors, each at 2 levels, that's going to give us, with a full factorial design, that's going to give us 64 runs. So 64 runs might sound like a lot of pipetting if you're doing it manually, but I promise if you've had your cornflakes, I'm sure you will be fine. But if that's really not possible, you could still do a fractional factorial design, which might give you 32 or even 16 runs. And with the 32 runs, you'd still be able to investigate all your 2-factor and main effects. So in summary, I'm saying there's no excuse. You should feasibly be able to investigate enough of your factors without getting any pet-related injuries and saving some cost.
Amazing. So you mentioned, I'll have to pick you up on it since you goaded me. So you mentioned doing buffer independently. Why do you think that might be safe in this case?
I think it's really about the fact of whether you're using your own knowledge here. And I think that in my experience, when you're changing, you might have 3 buffers you're interested in and you're happy to use that as a factor. In actual fact, you're not often getting much interaction between the buffer and the components. And so therefore, the reason you might want to bundle everything together, the reason you're doing DOE is because you want to account for these interaction effects. So you're interested in how everything impacts your response, but also how they impact each other to impact the response. And if you're pretty sure that buffer doesn't do that, you could investigate it separately. And I think that's a sweeping statement, but I think in this case, where the context is purposely being brutal, I think maybe you could afford to do it.
Yeah, it makes sense to me. I'll take it for it anyway.
Nice. Cool. All right, Sid. So what is multiple comparison testing and when should I do it?
Okay, love this question. It's one of my favorite hobby horses. I'm going to have to be a bit brief on this, and I'm afraid it is less application-y, but it's still a great topic. So I hope you enjoy it. So yeah, also, I'm very happy to say I get to use an XKCD cartoon. So Randall Munro's done the hard work here, which is brilliant. So yeah, just bear with me. So the setup is that we've been heard that jelly beans cause acne, and scientists investigate, do a significance test at 5% and say, no, there's no link. Then somebody comes back and says, no, I hear it's actually a specific color. Oh, all right, go on then. We'll try all the colors. And so they do. And they have 20 different colors to try. And lo and behold, green jelly beans, exactly one of the 20, come out as statistically significant as having a causal relationship with or association with more acne. So, of course, we get the news. Green jelly beans are linked to acne at a 95% confidence. Amazing. Okay, what's wrong with this picture? I hope it's pretty clear. Like, it doesn't take a lot to realize that 5% is 1 in 20. So purely by chance, we would expect one of these 20 significance tests to come up with a positive result. That's actually essentially what we've guaranteed by choosing a 5% threshold. Yeah, the situation in DOE where this arises most is obviously screening, where potentially you're covering a huge space of possible effects, and you're effectively simultaneously testing all these different hypotheses that each effect is significant. Which means if you do that all as a single fixed threshold of 5%, you're likely to exceed 20 pretty quickly. So what can you do about this? So there's a couple of corrections that are established and you'll see in most stats software. So the most common, the most easy to understand is the Bonferroni correction, where you take your original threshold and then you just divide it by the number of tests you're doing, and that gives you a new threshold. And this is known as controlling the family-wise error rate. So it just means you consider all your tests as being from the same family, so to speak, and you just guarantee that at most one of them or will have your false positive, essentially. And this is great. It's easy to understand. On the other hand, though, it's extremely stringent. It's very easy to accidentally throw stuff out this way. And I think, building on what I said before, and just generally, the context of a DOE is very different to the context of what we saw before, right? We're not trying to then make headline news and change the way, incur loads of costs on some poor manufacturers somewhere. All we're doing usually is deciding what we're going to test next, which is much lower cost. So being this stringent probably doesn't make an awful lot of sense. Instead, we might choose something like the false discovery rate, which is an alternative where, in this case, what we do is we think about, okay, so if we accidentally say something that isn't really significant, isn't really active, actually is, that's what we call a false positive, right? Whereas if we actually find something that's active and say it's active, that's a true positive. And so you basically, what you're trying to do here is just control how many false positives, what rate false positives at. So you're now setting this to something like, say, 5%. And that means you'll let a lot more of the true positives through as well in most cases. And that helps you to then be a lot more sensitive while still not letting too many incorrect things through. And that's usually the right sort of approach for something like DOE. And you'll usually see this in most packages. So just to hammer home the point about screening, this is an example of a screen with quite a lot of different factors and effects in it. As you can see, I think there's 72 here. So we're doing 72 possible tests. So yeah, 5%, we'd see quite a few just come up at random. That's probably not too bad, but we might still be a bit concerned. And I think that the key point here is the reason you're going to be concerned is you're going to end up with a model that fits far too well to the data and is actually starting to be sensitive to noise and isn't really telling you stuff. And that can happen particularly in bigger DOEs where you've got a lot of runs. So really, I tend to be a bit flexible at how I do this. I tend to mostly default to using the Bonferroni, actually, because a lot of DOEs do tend to, you see the difference between the individual and the simultaneous here. It's like there's a lot more effects on the left, and that probably would give you a much too accurate model given the quality of the data. The one on the right with sort of 7 effects, probably a lot better. It's usually a good choice, especially when you see this sort of situation, we see millions of them. It's not so good in other cases where you see very few significant effects, and there you might want to be a bit more liberal about it. So yeah, in summary, it applies anytime you're searching for a model. The original question asked about the response surface stage. At that point, I wouldn't typically tend to think about doing this in more classical approaches, but in the modern world, we do tend to combine screening and optimization, so it can apply. But you've just got to think about the risk you're taking. Adding a couple of extra terms in a model probably isn't the worst thing you can do. So I would typically tend not to use it so much. But yeah, it depends on your situation. Just don't blindly follow the statistics. All right, I think we better barrel straight on into this next one, actually. So we've got 5 minutes left. So yeah, should be a short one, though. Do you have an example case study of DOE assay robustness, Nathan?
Yeah, I feel personally attacked by your last bullet point. Don't blindly follow the statistics. Yeah, this is one of those questions that's so perfect it looks planted. I promise it isn't, and I'll give an example. The specific question did ask for ELISA examples. I don't have any. We do have a case study that's going to cover assay robustness, though. I'll do my best to cover that. And this was actually run by one of our lab scientists at Synthace for their thesis at University College London. And they recognize that assay development is a particularly good application of DOE. You've got an assay for a complex biological process. You want to optimize its signal. You're probably under massive pressure from the team who want to use your assay. And I'll link the case study when we upload this recording. But in short, they took 12 factors and they were tested across 3,456 runs, which was 20,745 liquid handling steps. And really, there is no amount of cornflakes or Weetabix or pre-workout that would give you enough energy to do that manually. So we were lucky. We had the Dragonfly dispenser by SBT Labtech, which I can assure you would have smashed through that very quickly. Before the DOE experts come at us saying that 3,456 runs is far too many for 12 factors, we're going to dig into why. But really the aim here is to demonstrate how a complex, high-dimensional environment can be characterized with one automated DOE, which is almost ironic because for the last half hour, we've been talking about iterations, scoping, screening, and this is an example where they didn't do much of that in depth intentionally. There was a two-part scoping study before this actually that determined some of the enzyme and buffer concentration levels, but really this was a test to see if we could put everything into one very extensive design and gain those huge insights with one experiment. So this actual experiment was an aim to optimize enzyme activity in an assay. They sampled these 12 factors that they found commonly impact enzyme activity in literature, and they used very wide ranges for levels. So this made 432 runs, including controls, and this was replicated 8 times due to the wide-ranging levels and the number of factors. And really, they wanted to get an accurate understanding of the noise in the design space, which we spoke about before. But in total, this is where you get the 3,456 runs, and that was over 36 96 well plates. And how long did it take? Under 7 minutes. It was 6 minutes, 40 seconds per plate, which is not for the whole design, that's per plate, but still very impressive. And to do this, they used a space-filling design. And I wanted to pause to talk about this because it's quite a unique design where you set an upper and a lower limit for a factor, and then the design samples that design space in evenly spread intervals between those limits. And these are really thorough. They'll cover a huge amount of the design space and give a really comprehensive understanding of how factor affects response. I suppose a problem is because they're sampling at upper and lower, multiple points really between your upper and lower limits, they're going to require a huge number of experimental runs. They're often expensive. I suppose another consideration is that actually each factor level is only actually replicated or sampled once, if you like. There's no partial replication in a space-filled design. And in most designs, you'll get a particular level of a factor appear multiple times, just in different combinations with other factors. But in a space-filled design, you don't get that because you're sampling at a range of levels between your upper and lower limit. So they're often used when you don't have maybe a prior knowledge of the experimental space, you're going in blind. But in terms of assay robustness, this example is great. There's often a need to cut down the number of factors you test. But when you've got a beast of a liquid handler or dispenser like we did, you're not really as constrained in terms of your experimental runs. Here, they really tested the lot and included all 12 factors that they found from the literature. And if you want your experiment to be robust, you're going to want to include factors that you want your system to be tolerant to. So in this case, and something we see a lot, is that people include something like DMSO as a factor. Even though it might not be something that they're interested in changing, they'll add it as a factor simply because they want to see if their conditions are DMSO tolerant. So, in actual fact, this can save you follow-up experiments like a DMSO tolerance test. And despite its faults, this space-filled design gave them huge coverage and a uniform coverage of the design space, which a constrained design might not give you. And those 8 replicates, you might sound like a lot, but really was able to account for that lack of partial replication that I spoke about, but also gave a really robust estimation of the noise in the system and actually allowed them to model the noise too. So yeah, check this one out. We'll leave it in the description. It's really cool.
Fabulous. Okay, I think we better summon Ross. Here he comes.
On cue.
Hello, cool. So thank you very much, guys. That was really interesting. We had some good interaction in the chat as well. So everybody, thank you for taking part. I know we are out of time, but I just wanted to very quickly share a couple of follow-up things that people may be interested in. So we do have a DOE training email course where you can learn DOE. I think there's 8 installations of this course, just 6 minutes a day. There's some really great insights in there. So I'd encourage everybody who hasn't already to sign up to that. I did then have a slide where we were going to pose some questions in the chat, but we are up on time. But I would just encourage everybody to reach out to us. I think we can share contact details. We're on LinkedIn. Drop us a message. We're more than happy to answer any sort of questions offline. And then just a final shout out just to say This is obviously the second installment of our DOE Office Hours series. We do have a third installment coming up at the beginning of October. For those of you who have registered, you should receive a reminder about this in the coming days. In that session, we'll be covering a little bit more on DOE, but focusing a little bit more on the Synthace platform itself. So if you'd like to hear about some of the tools and functionalities we've developed for allowing people to do DOE, then that really is the time to come along and attend. And with that, I guess, thank you to Sid and Nathan today. It's been a pleasure to hear your insights, and thank you very much, everybody, for attending.
Thanks, Ross. Thanks, everyone.
See you later. Bye.
Cheers.